Genes are sections or segments of DNA that are present on chromosomes. These genes determine characteristics in living organisms. Passing of genes from one generation to the next is called heredity. Genes are made out of DNA, a chemical code with 4 different letters in the form of bases. Every time a cell divides, it must copy, or we may say double its DNA, so that each of the 2 daughter cells receives 1 copy each of the parent DNA. A cell may make some errors when it copies its DNA. Most of these are corrected immediately, but a few manage to escape unnoticed. These uncorrected errors cause mutations in the cells. Mutations are either inherited or acquired during one's lifetime, as one's body cells undergo aging or are exposed to certain chemicals.
ਵਿਦਿਆਰਥੀਓ, ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਤੁਹਾਡਾ ਸਵਾਗਤ ਹੈ। ਲਿਵਿੰਗ ਆਰਗੇਨਿਜ਼ਮਸ ਯਾਨੀ ਸਜੀਵ ਸੈੱਲਾਂ ਤੋਂ ਬਣੇ ਹੁੰਦੇ ਹਨ। ਇਨ੍ਹਾਂ ਸੈੱਲਾਂ ਵਿੱਚ ਇੱਕ ਪਦਾਰਥ ਹੁੰਦਾ ਹੈ ਜਿਸ ਨੂੰ ਡੀਆਕਸੀਰਾਈਬੋਨਿਊਕਲੀਇਕ ਐਸਿਡ ਜਾਂ DNA ਕਿਹਾ ਜਾਂਦਾ ਹੈ। DNA ਮਿਲ ਕੇ ਕ੍ਰੋਮੋਸੋਮਸ ਬਣਾਉਂਦੇ ਹਨ। ਆਮ ਤੌਰ ਤੇ ਮਨੁੱਖੀ ਸਰੀਰ ਦੇ ਸੈੱਲਾਂ ਵਿੱਚ ਕ੍ਰੋਮੋਸੋਮਸ ਦੇ 23 ਜੋੜੇ ਹੁੰਦੇ ਹਨ। ਮਤਲਬ ਕੁੱਲ ਮਿਲਾ ਕੇ ਇਨ੍ਹਾਂ ਦੀ ਗਿਣਤੀ 46 ਹੁੰਦੀ ਹੈ। Students, you know that each male and female gamete has unpaired 23 chromosomes. Humans receive half of chromosomes from their mother and half from their father. हर एक organism ਦਾ ਹਰ ਇੱਕ ਗੁਣ ਉਸ ਦੇ chromosome ਵਿੱਚ present DNA ਉੱਪਰ ਮੌਜੂਦ genes ਰਾਹੀਂ ਕੰਟਰੋਲ ਹੁੰਦਾ ਹੈ। So, DNA is a hereditary material that is transmitted from one generation to another without any change. ਪਰ ਕਈ ਵਾਰ genes ਵਿੱਚ changes ਵੀ notice ਕੀਤੀਆਂ ਗਈਆਂ ਹਨ। ਇਨ੍ਹਾਂ changes ਨੂੰ genes ਦਾ disorder ਕਿਹਾ ਜਾਂਦਾ ਹੈ। So, let us see what are these genetic disorders. Sometimes, cells undergo changes in the set of information present in them. These are called mutations, and these mutations often occur when cells undergo aging or have been exposed to certain chemicals or radiations. Fortunately, cells usually recognize these changes and correction takes place almost naturally. But sometimes, these changes or mutations may cause certain serious diseases like hemophilia, sickle cell anemia, and some types of cancer. Genetic disorders can be defined as a disease that is caused by any abnormality in an individual's DNA. Abnormalities can range from a small mutation in a single gene to addition or subtraction of one chromosome or a set of chromosomes. One of the most important threats to human health these days is posed by genetic diseases. Researchers have defined more than 4,000 diseases that are caused by genetic variations. Some genetic disorders are more likely to occur if one has a family history of such disorders. These disorders are transmitted to the offspring, and the pattern of inheritance of such disorders can be traced in a family by the pedigree analysis. A pedigree is a diagram of family relationships that uses symbols to represent people and lines to represent genetic relationships. Pedigree analysis is done to check the inheritance of characters from parents and grandparents. It would be more clear with visuals.
A pedigree is a record of inheritance of certain genetic traits for 2 or more generations, presented in the form of a diagram. Parents are shown by these geometrical figures: square for male and circle for female. They are shown connected by horizontal line. Their offsprings are shown connected by a vertical line. The offsprings are numbered with Arabic numerals, whereas generations are numbered with Roman numerals. Solid symbols represent traits under study. Once the family members are properly arranged, important medical facts can be added. Once the inheritance pattern of any genetic disorder is determined, the status of family members in the pedigree can be evaluated. It is used by the genetic counselors to advise couples about the possibility of having children with genetic defects.
So students, from pedigree analysis, the risk of carrier status for other family members or the chance that a couple may have an affected child can be estimated. Pedigree analysis provides a strong tool which is utilized to trace the inheritance of a specific trait, an abnormality, or any genetic disease. But the real question here is that how these genetic disorders are inherited. Our body is made up of trillions of cells. Each cell has a core structure, that is, nucleus, that contains our chromosomes. Chromosomes are made up of DNA and some proteins. A gene mutation is an alteration in one's DNA. Some changes in our genes may result in genetic disorders. ਜੇ ਮਨੁੱਖੀ genetic material ਦੇ ਵਿੱਚ ਕੋਈ ਖਰਾਬੀ ਆ ਜਾਵੇ ਤਾਂ ਉਹ ਮਨੁੱਖ genetic disorder ਦਾ ਸ਼ਿਕਾਰ ਹੋ ਜਾਂਦਾ ਹੈ। ਸਾਰੇ genetic disorders genes ਜਾਂ chromosomes ਵਿੱਚ ਕੋਈ ਨਾ ਕੋਈ ਖਰਾਬੀ ਹੋਣ ਦੇ ਕਾਰਨ ਹੁੰਦੇ ਹਨ। ਪੀੜ੍ਹੀ ਦਰ ਪੀੜ੍ਹੀ ਚੱਲਣ ਵਾਲੀਆਂ ਬਿਮਾਰੀਆਂ genes ਵਿੱਚ ਆਈ ਖਰਾਬੀ ਦੇ ਕਾਰਨ ਹੀ ਹੁੰਦੀਆਂ ਹਨ। These disorders can be dominant, recessive, or sex-linked. Let us study the types of genetic disorders. There are two main types of genetic disorders: chromosomal disorders, gene-related disorders. Let us start with chromosomal disorders. Chromosomes hold the genetic keys to all the functions of body. There may be errors in any of the body's 23 pairs of chromosomes, such as broken or missing pieces, or too many pieces, or multiple copies. These chromosomal abnormalities can result in genetic diseases. Down syndrome, Klinefelter syndrome, and Turner syndrome are examples of chromosomal disorders. Down syndrome is a developmental disorder caused by an extra copy of chromosome 21. That is why the disorder is also called trisomy 21. Having an extra copy of this chromosome means that each gene may be producing more protein product than normal. Genes on chromosome 21 that specifically contribute to various symptoms of Down syndrome are now being identified. Let us know something more about Down syndrome.
Down's syndrome. Down's syndrome is typically caused by what is called non-disjunction during oogenesis. If a pair of chromosome number 21 fails to separate during the formation of an egg, this is referred to as non-disjunction. When that egg unites with a normal sperm to form an embryo, that embryo ends up with 3 copies of chromosome 21 instead of the normal 2. This extra chromosome is then copied in every cell of the baby's body. An extra chromosome 21 for a total of 47 chromosomes instead of 46. Symptoms of Down syndrome. People with Down syndrome have very distinct facial features, decreased muscle tone at birth, abnormally shaped face, a small broad nose, partially open mouth with protruding tongue, slanting eyes with small folds of skin in the corners.
ਡਾਊਨ ਸਿੰਡਰੋਮ ਦੇ ਰੋਗਾਂ ਵਿੱਚ ਕਈ ਤਰ੍ਹਾਂ ਦੀ ਮੈਡੀਕਲ ਪ੍ਰੋਬਲਮਸ ਦੀ ਆਸ਼ੰਕਾ ਵੱਧ ਜਾਂਦੀ ਹੈ, ਜਿਵੇਂ ਕਿ ਰੈਸਪੀਰੇਟਰੀ ਇਨਫੈਕਸ਼ਨਸ, ਬਲੌਕਡ ਡਾਈਜੈਸਟਿਵ ਟ੍ਰੈਕਟ, ल्यूकीमिया, हार्ट डिफेक्ट्स, hearing loss, hypothyroidism, and various eye abnormalities, etc. Chromosomal abnormality ਦੇ ਕਾਰਨ ਹੋਣ ਵਾਲੀ ਸਭ ਤੋਂ ਆਮ ਬਿਮਾਰੀ ਡਾਊਨ ਸਿੰਡਰੋਮ ਹੈ। ਇਹ ਵੇਖਿਆ ਗਿਆ ਹੈ ਕਿ 800 ਤੋਂ 1000 ਬੱਚਿਆਂ ਵਿੱਚੋਂ ਕੋਈ ਇੱਕ ਇਸ ਬਿਮਾਰੀ ਤੋਂ ਪੀੜਿਤ ਹੁੰਦਾ ਹੈ। ਹੁਣ ਤੱਕ ਡਾਊਨ ਸਿੰਡਰੋਮ ਦਾ ਇਲਾਜ ਸੰਭਵ ਨਹੀਂ ਹੋ ਸਕਿਆ ਹੈ, ਪਰ ਫਿਜ਼ੀਕਲ ਥੈਰੇਪੀ ਅਤੇ ਸਪੀਚ ਥੈਰੇਪੀ ਕੁਝ ਹੱਦ ਤੱਕ ਇਸ ਬਿਮਾਰੀ ਨਾਲ ਪ੍ਰਭਾਵਿਤ ਲੋਕਾਂ ਦੀ ਸਹਾਇਤਾ ਕਰ ਸਕਦੀ ਹੈ। ਇਸ ਤੋਂ ਵੱਧ ਜੇ ਅਜਿਹੇ ਲੋਕਾਂ ਨੂੰ ਉਚਿਤ ਵਾਤਾਵਰਣ ਦਿੱਤਾ ਜਾਵੇ ਤਾਂ ਉਹਨਾਂ ਦੀ ਸਿੱਖਣ ਦੀ ਸਖਿਅਮਤਾ ਵੱਧ ਸਕਦੀ ਹੈ। Next chromosomal disorder is Klinefelter syndrome, which affects males only. Students, you know males have an X chromosome and a Y chromosome XY. But males having Klinefelter syndrome have an extra X chromosome, means they have a XXY chromosome instead of XY. This gives them a total of 47 instead of the normal 46 chromosomes. Let us have a look on this type of syndrome.
Klinefelter's syndrome. Klinefelter's syndrome is a condition that occurs in men as a result of an extra X chromosome. The affected males are sterile. Normally, males inherit one X chromosome from their mother and one Y chromosome from their father. But those who have Klinefelter syndrome inherit an extra X chromosome. Klinefelter syndrome is typically caused by what is called non-disjunction, in which an abnormal egg with XX chromosome unites with a normal sperm to form an embryo. That embryo may end up with 3 copies of the 6 chromosomes, that is, XXY, instead of the normal 2, that is, XY. The extra chromosome is then copied in every cell of the baby's body. Males who have Klinefelter syndrome may have the symptoms such as small, undeveloped testes which don't make enough testosterone. Therefore, these males have sparse armpit and facial hair, enlarged breasts, tall stature, or we may say abnormal body proportions, that is, long legs and short trunk. Women who have pregnancies after the age of 35 have a slightly increased chance of having a baby with this syndrome.
Klinefelter syndrome is one of the most common genetic abnormalities. Klinefelter syndrome is found in about 1 out of every 500 to 1,000 newborn males. The disorder is named after Dr. Harry Klinefelter, who first reported its symptoms in 1942. Next disorder that we want to discuss today is Turner syndrome. Turner syndrome is a medical disorder that affects about 1 in every 2,500 girls. Turner syndrome is caused by the presence of missing incomplete sex chromosomes. So, Turner syndrome at times leaves her with only one fully functional X chromosome. The genes affected are involved in growth and sexual development. That is why girls with this disorder are shorter than normal and have abnormal sexual characteristics.
How do people get Turner's syndrome? Generally, girls are born with 2 X chromosomes as sex chromosomes, but girls with Turner's syndrome are born with only 1 X chromosome. The symptoms of this condition vary widely among girls with Turner's syndrome. It all depends on how many of the body cells are affected by the changes in X chromosome. Girls with Turner's syndrome usually have rudimentary ovaries, undeveloped breasts. As a result, one tends to be sexually underdeveloped and infertile. They are usually of short stature. Short stature is accompanied by webbed neck, puffy fingers, abnormal fingernails and toenails.
The best thing is that when Turner syndrome is diagnosed while a girl is still growing, she can be treated with growth hormones to help her grow taller. As you know that there are 2 main types of genetic disorders: chromosomal disorders and gene-related disorders. ਅਸੀਂ chromosomal disorders ਦੀਆਂ ਕਿਸਮਾਂ ਬਾਰੇ ਗੱਲ ਕਰ ਚੁੱਕੇ ਹਾਂ ਅਤੇ ਹੁਣ ਅਸੀਂ gene-related disorders ਬਾਰੇ ਚਰਚਾ ਕਰਾਂਗੇ। ਇਹ ਸਾਰੇ disorders single mutated gene ਦਾ ਹੀ ਨਤੀਜਾ ਹੁੰਦੇ ਹਨ। Gene defects ਦੇ ਕਾਰਨ ਲਗਭਗ 4000 ਤੋਂ ਵੱਧ ਮਨੁੱਖੀ ਬਿਮਾਰੀਆਂ ਹੋ ਸਕਦੀਆਂ ਹਨ। ਅਕਸਰ ਇਹ ਵੇਖਿਆ ਗਿਆ ਹੈ ਕਿ ਸਿੰਗਲ ਜੀਨ ਡਿਸਆਰਡਰਸ ਦੇ ਕਾਰਨ ਇਹ ਜੈਨੇਟਿਕ ਡਿਸਆਰਡਰਸ ਸੰਬੰਧਿਤ ਪੀੜ੍ਹੀਆਂ ਵਿੱਚ ਅੱਗੇ ਤੋਂ ਅੱਗੇ ਪਾਸ ਹੁੰਦੇ ਰਹਿੰਦੇ ਹਨ। ਇਹ 2 ਕਿਸਮਾਂ ਦੇ ਹਨ: ਆਟੋਸੋਮਲ ਰਿਸੈਸਿਵ ਐਂਡ ਆਟੋਸੋਮਲ ਡੋਮੀਨੈਂਟ। Autosomal recessive disorders are caused by recessive autosomal genes, means 2 copies of an abnormal gene must be present in order for the disease or trait to develop. Autosomal dominant disorders are caused by dominant autosomal genes, means a person needs to get the abnormal gene from one parent in order to inherit the disease. One of the parents may often have the disease. Let us discuss some autosomal recessive disorders: hemophilia, sickle cell anemia, and phenylketonuria. Let us discuss one by one. Hemophilia is a disease in which a patient shows abnormal blood condition in which bleeding from a cut or injury does not stop. The bleeding occurs because the blood does not clot. A person with a small cut or internal bleeding could bleed to death. The most prevalent symptom of the disease is continuous bleeding in case of injury or bruising. In some instances, the bleeding can occur without any external reason. While wounds do not pose much risk in the affected people, internal bleeding can have fatal consequences. ਇੱਕ ਹੋਰ autosomal recessive disorder, sickle cell anemia ਨੂੰ visual ਰਾਹੀਂ ਸਮਝਦੇ ਹਾਂ।
Sickle cell anemia is a disorder that affects red blood cells which use a protein called hemoglobin to transport oxygen from the lungs to the rest of the body. Normally, red blood cells are round and flexible, so they can travel freely through narrow blood vessels. The hemoglobin molecule has two parts, an alpha and a beta. Patients with sickle cell anemia have a mutation in a gene on chromosome number 11 that codes for the beta subunit of the hemoglobin protein. As a result, hemoglobin molecules become affected, causing red blood cells to be rigid and have a concave shape like a sickle. These cells don't move easily through blood vessels. The clumps of sickle cells block blood flow in the blood vessels, may cause pain, serious infections, and organ damage. Other symptoms include delayed growth, strokes, and jaundice. Life of these cells is less than normal RBCs. As a result, people with this disorder often have low red blood cell count, that is anemia, which is why this disease is commonly referred to as sickle cell anemia.
Students, you know because of the changed shape of RBCs, this disease prevents oxygen from reaching the spleen, liver, kidneys, lungs, heart, or other organs, causing a lot of damage. Without oxygen, these organs begin to die. Sickle cell anemia is particularly common among people whose ancestors came from sub-Saharan Africa, India, Saudi Arabia, and Mediterranean countries. Next disorder is phenylketonuria, that is PKU. Phenylketonuria is a rare metabolic disorder that affects the way the body breaks down protein. If not treated shortly after birth, phenylketonuria can be destructive for nervous system, causing mental retardation.
Phenylketonuria is caused by a mutation in a gene on chromosome number 12. The gene codes for synthesis of a protein called phenylalanine hydroxylase, an enzyme present in the liver. This enzyme breaks down the amino acid phenylalanine into tyrosine. When the gene is mutated, the phenylalanine hydroxylase enzyme changes its form in a way which is unable to break down phenylalanine into tyrosine. Phenylalanine builds up in the blood and poisons nerve cells in the brain. Babies born with PKU usually have no symptoms at first, but if the disease is left untreated, babies experience severe brain damage. This damage can cause epilepsy, behavioral problems, and arrest the growth of the baby.
PKU, that is Phenylketonuria, is an autosomal recessive disorder, meaning that you need to inherit mutations in both copies of the gene to develop the symptoms of the disorder. So students, this was all about genetic disorders. With this, we conclude our lesson for today. Let us revise what we have learned so far. A genetic disorder is a disease that is caused by an abnormality in an individual's DNA. There are two main types of genetic disorders: chromosome-related disorders and gene-related disorders. Chromosomal disorders are the errors on any of the body's 23 pairs of chromosomes, such as broken or missing pieces or too many pieces or copies. The examples of chromosomal disorders are Down syndrome, Klinefelter syndrome, and Turner syndrome. Gene-related disorders are the result of a single mutated gene. There are estimated to be over 4,000 human diseases caused by gene defects. Common gene-related disorders which we discussed today are hemophilia, sickle cell anemia, and phenylketonuria. Okay, now let me check how much you have grasped. I will ask you some questions and I hope you will answer them. The first question is, what do you understand by the pedigree analysis? Answer: a pedigree is a diagram to represent genetic relationships. Pedigree analysis is done to check the inheritance of characters from parents and grandparents. It uses symbols to represent people and lines to represent genetic relationships. Next question. What causes Down syndrome? Answer: Down syndrome is caused by a condition which is called as non-disjunction. If a pair of chromosomes number 21 fails to separate during the formation of an egg, this is referred to as non-disjunction. When that egg unites with a normal sperm to form an embryo, that embryo ends up with 3 copies of chromosome 21 instead of the normal 2. My next question is, what are the two main types of genetic disorders? The answer is, there are two main types of genetic disorders: chromosomal disorders and gene-related disorders. The next question is, these are the examples of genetic disorders. Classify them in chromosomal and gene-related disorders. The options are Down syndrome, sickle cell disease, Turner syndrome, and phenylketonuria. Answer: out of these, Down syndrome and Turner syndrome are chromosomal disorders, whereas sickle cell disease and phenylketonuria are gene-related disorders. So students, this was all about genetic disorders. This concludes our today's lesson. Thanks for your cooperation. See you next time. Goodbye.